Hello, everyone, and welcome to today's live broadcast. The revised ISO 14644-1 changes classification and monitoring methods. Are you prepared? I am Suzanne of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. This webinar is sponsored by Beckman Coulter. Beckman Coulter Life Sciences is dedicated to empowering discovery and scientific breakthroughs. The company's global leadership and world-class service and support delivers sophisticated instrument systems, reagents, and services to life sciences researchers in academic and commercial laboratories, enabling new discoveries in biology-based research and development. A leader in centrifugation and flow cytometry, Beckman Coulter has long been an innovator in particle characterization and laboratory automation, and its products are used at the forefront of important areas of investigation, including genomics and proteomics. Before we start, there are some few instructions. We want to hear from you during this interactive broadcast, so please ask questions or leave us a comment. Answers are also welcome. You can do this by hitting the green Q&A button at the lower left of the presentation window and typing in your comments and questions. We're trying to get to as many questions as we can, and we'll follow up with you if we don't have time today. Do you want a better look? You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you can't see or hear this presentation properly, let us know by clicking on the support button at the top right or use the Q&A button. We'll make sure to resolve any issues. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located at the bottom left-hand corner of your webpage and follow the process of obtaining your credit. Now let's get right to today's speakers. We're proud to welcome Joe Gecsey. Joe has been involved with the particle counting industry since 1984. He first joined Met1 as an electronic design engineer and has since been involved in both airborne and liquid particle counter design, as well as multi-sensor systems for facility monitoring systems. Joe has been involved in various standards involving particle counting in air and liquid. He was one of the 2 Americans on the ISO 14644-1 revision committee and headed the subcommittee to redraft the ISO 14644-2 standard on monitoring of clean rooms. Currently, Joe is the Life Science Application Manager at Beckman Coulter Life Sciences, Particle Counting and Characterization Division. I will now turn it over to Joe for his presentation.
Thank you, Susan. Hello to everyone, and thank you for joining us today. The topic today is on the classification methods, so we'll dive into that and where some of the changes recently may affect what you do in the daily efforts for monitoring clean rooms, whether it's strictly pharmaceutical, biotech kind of thing with looking at the what you would call normally EM or environmental monitoring, or something broader to do with clean rooms in general. So try and look at the various types of application, but mainly focus towards the pharmaceutical and biopharmaceutical world, because that gets to be a little bit more interesting and tricky at times. So just as a little bit of background, before the ISO 14644 existed, most countries had their own type of monitoring standard. In the U.S. it was the Federal Standard 209, and a lot of confusion and mixed efforts affected people being able to consider how the clean room should be organized, especially internationally. So in 1999, the ISO 14644 was created with the first couple of sections, -1 and -2, related to the level of airborne particles in the air, and being able to look at the concentration in the air and make sure it's appropriate for various purposes. It's important to realize that this standard affects or is used by all industries. It's not just a pharmaceutical or medical device or biopharmaceutical standard. It really relates to anyone working in a clean space and wanting to be able to understand the level and concentration of the particles in the air, and again, knowing that it's appropriate for their particular use, it's clean enough. There are more than just the two sections for this standard. In fact, today there's 15 of them. We're going to focus on the first two, again, basically looking at the concentration of airborne particles. There are other ones, especially some new ones related to nanotechnology that may be worth looking into, should that be an area of need. But fundamentally, 1 and 2 are our focus and relate to airborne concentration. One of the things that's very important to understand, and I hope everyone leaves today, this webinar with this a clear understanding, even though we have instruments, particle counters that are used throughout the applications here, there's really two fundamentally different philosophical, you could say, uses for it. And one of those is what we're talking about really today, focused on, that is classification. And the other is a monitoring application. That is something where you're testing more frequently, often on a daily or even continuous basis. And these are really two quite different applications, but often using exactly the same instrument. So hopefully, as we go through this discussion today, that will become clear, and the appropriate use of the standard for classification versus other uses of different documents and procedures for monitoring will become a bit more distinct and clear. So just as a general discussion, when we look at these two types of uses, that is, looking at classification and looking at monitoring, the frequency is the first thing that is significant. Basically, classification is a formal study of an area done maybe every 6 months or every 12 months. There are formulas for setting up the number of positions and the sample volume. There is a singular pass-fail criteria. You either meet the class or don't. There's a fairly standardized format for making the report. And it's somewhat assumed that the area room is fairly uniform or homogeneous in terms of the distribution of particles in the air. Whereas when you look at the monitoring side, you're really dealing with something that is done more frequently, and you're trying to understand the current health, you might say, of the area. Is it still appropriate for the use? And in that way, there's no formula for that. It's mainly a matter of looking at the data that is needed to understand that level of concentration of particles. Same with the sample volume. It's really defined by the need for information, the sense of risk, perhaps. And there are often controls set up that are based on commonly called alert and action levels. There's a couple of levels, and they're often either much lower than the classification used for the room, or in some cases higher even than that. So they're unique and not necessarily related to the class limit value that's set up in classification. And generally the report form takes something like a quick graph or other summary, so it's a matter of understanding quickly what that particular area is at for concentration, and again, that it meets the current need. One of the key differences with monitoring is that each sample position has some unique characteristics, and it doesn't necessarily relate to the rest of the zone. You could have one position that's extremely clean, maybe even located in front of a filter or underneath a filter area, whereas you may have another one where operations occur or people pass by, even though it's in the same considered zone or room. It will have a different characteristic or signature of counts of particles in the air. And so everything is unique when you start looking at monitoring in terms of the position. Whereas for classification, we're really thinking about the whole zone, area, room, biological safety cabinet, bench, whatever that zone or area might be. So keep these in mind when we talk about the differences. We focus, as I said, on classification, but we'll also touch on the needs for monitoring as we go through this discussion today. So classification, monitoring, two distinct approaches to looking at concentrations in the air. So again, when we look at classification, the room and zone, as we'll see, is in this closer look at the standard. Monitoring, each sample position becomes the focus of the assessment. So let's look particularly at the classification function for airborne counts. What is the standard that we're addressing here, this ISO 14644? Its main purpose is two things. First thing you could say is defining the cleanroom class, being able to have a common language. I can speak to someone about an ISO class 5 room, and they'll have a sense of what that means. Maybe it'll bring up a mental image of someone dressed up in a complete garment, a full bunny suit, as we would say, with even goggles on. Whereas you might say an ISO 8, maybe it's much more relaxed, just a lab coat. ISO 2, okay, something maybe even fully gowned, not only just fully gowned, but in a somewhat of a fully enclosed spacesuit type. So it helps again with the understanding, especially across industries or internationally, for the level of cleanliness that's intended for a zone or area. It also helps put forward a statistical sampling method for that room. And it's often something people forget or don't realize at first when you're doing the classification effort. We don't have 100% confidence of how that zone is. We haven't been able to extract 100% of the air at any given moment to know precisely what the concentration is in that whole zone. So instead we use a statistical sampling method, basically choosing points within the area and a certain amount of sample volume at each of those points, using that data set to extrapolate our confidence that the whole area looks similar to that data set or not. And so really it's defining a statistical sampling method, looking at number of points, so it sets up a minimum number of points, and how much air needs to be sampled at each point to give us a high level of confidence that that data set is going to represent the whole entire area or room. So currently, this is a chart you would find, well, I should say currently in the 1999 version, which was current up until December. And this table basically defines the ISO classes 1 through 9 and the 6 different sizes that you can use as standard to define those rooms. You don't have to use all sizes for a particular zone, just the ones of interest. For the life science industry, for example, in ISO 5, mainly it's 0.5 micron and 5 micron, whereas for some other operations, maybe for working with space vehicles, maybe you might be interested at ISO 5, but at 0.3 or even 0.1 microns. So it's really dependent upon the application at hand, how to employ the chart for your particular operation, but defines the upper limit in terms of the concentration of particles per cubic meter. This is a maximum concentration. So, for example, it's at ISO 5 at 0.5 micron. The maximum concentration at 0.5 micron must be no greater than 3,520 at each position, or you cannot consider it an ISO 5 class room. So that's basically defining a pass-fail for a given size and ISO class. These classes are very similar to those that people may have experienced before from the federal standard, U.S. federal standard. Class 100, 10,000, 100,000 were all based on counts per cubic foot. So it was a different measurement criteria, counts per cubic foot versus counts per cubic meter. There's 35.31 cubic feet per cubic meter, so this 100 times 35.31 basically accounts at 0.5 micron to the same number, similar for 10,000, 100,000. So the values did not change extremely for these particular zones, and there is somewhat of a connection between the old federal standard and the current 14644, just in terms of cubic feet in the federal standard, cubic meters when we talk about the ISO. It is important to recall, too, from the standard, there is no need to actually sample a cubic meter according to ISO. There may be other reasons to do that for other types of classifications, but in ISO, it's a matter of resolving the counts that you've taken to the equivalent counts per cubic meter. It's not demanding that a full cubic meter be taken. So this standard, which has been in place since 1999, almost it's more than 15 years, being used in the industry quite effectively to help people understand the capability of an area or room and that it's appropriate to their use. Finally, went through the revision process, and a effective date of December for this revision was set. So this is now officially revised. So we call it ISO 14644-1:2015. So what's changing? Well, it didn't radically change, and it was part of the effort of the group to make it somewhat consistent with the historical values, and so the people's classifications would not be altered significantly, if at all, by any of the changes made. So we tried to be quite consistent, but try to provide a better statistical base for the assessment. So in doing that, a few things we'll point to as we look at this, and I'll just talk about really two of the major ones, the ones that really might have an effect on how you perform classifications. So in terms of things that are changing in particular, there was a need to calculate an upper confidence limit value in the previous document for sample areas where you took a small number of space locations, less than 10. You had to do one further step. That requirement has been withdrawn in the new revision. The frequencies of retesting used to be specified by class. If it was ISO class 5 or cleaner, this period was 6 months, and if it was 6, 7, 8, 9, it could be 1 year. That's no longer delineated by class. And the calibration method and the number of locations, those things all changed. What didn't change, classes are still basically labeled the same way, ISO class 1 through 9. The values in most cases are all identical. We use the same equation for the calculation of sample volume, and there's still a minimum sample time of 1 minute. Those things all stayed the same. So these are the four kind of significant things, but two of these, that is the calibration of equipment and a minimum number of locations, are perhaps the more significant and meaningful ones, so we'll focus on those. Again, the new chart here, based on 14644-1:2015, looks almost identical, other than the outlying values here have been, in most cases, removed for reasons of statistical accuracy. So if we look again at these two things, we'll focus on that, the calibration equipment, minimum number of locations. So the first major change, and I think the one that's most significant to people, minimum number of locations, we've increased those in most cases so that the statistical validity of the sample set is improved. We're trying to find something that gives 95% confidence that the data set you've taken can be extended to the whole room. So previously this was determined by taking the square root of the measurement area based in square meters. It's now replaced with a lookup chart, and this defines the minimum sample locations. Those can be increased. You can do beyond that, especially if you have certain high-risk areas you wish to have good confidence about. So in most cases that means a small increase in the number of sample points. So let's look at that chart. So this is located in the appendix. For some reason I'm not seeing the chart display. One moment, please. Okay. So, all right. So if we look at the chart, the important thing we notice here is, for example, in the sizing, the numbers for most sizes will have increased. For example, if we took 36 square meters as an area that we're interested in defining minimum number of sample locations, in the previous method, we would have said, take the square root of that area in square meters, and that will define the minimum number. Well, square root of 36 would be 6. However, the chart today says the number needs to be 9. And again, this is based on the effort to try to make a stronger statistical platform to make our decision that, yes, the room meets that criteria with reasonable confidence, with 95% confidence of that. So you'll find in most cases similar modest increases in the number of points spread throughout the area. The second thing that's important is the change to the calibration method. There always was a specification that said the unit, the instrument used to execute the classification needed to have a current calibration, but how that calibration was performed was not necessarily defined. In the revision, a specific method, the ISO 21501-4, was specified, and this has certain positive effects. First off, in the previous methods, 4 different typical things were checked. In the ISO 21501-4, we really look at more than just those, and importantly, we look at counting efficiency of the particular instrument involved. This gains us a more consistent health check, you could say, of the instrument throughout its lifetime, and also more consistency between instruments. So in the today's method with 21501-4, we really improve the overall use of the instrument and make that mandatory, so we know we have a good, solid tool to execute the calibration. So we get some consistency, we remove ambiguity between someone doing calibration with method A and someone else doing it with method B. We're basically saying use the 21501. And this calibration standard, by the way, is not new. It's actually been there since 2007. And so it's something that's familiar generally to the industry. And what that gains us is much more consistent performance throughout the lifetime of the instrument and better unit-to-unit reproducibility. So our overall counting accuracy, and based on that counting efficiency test, improves. The downside, of course, it takes a little bit longer to do that calibration, takes more effort, and generally will somewhat increase the cost of that. But it does gain something for us in terms of the consistency of the instrument. And when you're setting up as a standard, that's important. You want it to be reproducible no matter which tool you use, which instrument you use, and who does it. So that's the idea, of course, of a standard. It's reproducible no matter who does it and if an appropriate instrument is always used. I want to mention one other tool that's hidden, if you want to say, inside of the standard. And this was actually there in the earlier standard, in the 1999 standard, and even in the federal standard. That often helps speed up the test for some applications, but not all, as we'll see. This is called sequential statistical sampling. It's an optional method. It's often very useful for clean areas. And again, because this is a statistical method, it's using statistics to foreshorten the total time taken. And when you're in very, very clean areas, ISO 5 and cleaner in most cases, then the sample times can start becoming long because, again, we want statistical confidence that the values we've taken fall within our categories, and often that takes a certain amount of time to verify that. However, because we're assuming certain consistency of the area over time, the sample point we're at, it doesn't change significantly over time, at least the time of the sampling. We're making a statistical assumption here in this method that we can quickly, by rate, figure out that we're going to pass or we're going to fail based on a few minutes of sampling. So this can be employed. It was before known as Annex F in the previous standard, and in the current one it's Annex D. Again, very much the same function and use as previously. And what it really speaks to is using the rate of counting to predict the likelihood of passing or failing classification. So again, it's trying to use the statistical methodology to help us move forward more quickly in this evaluation. However, it's very important to realize that if you're working with the European or the PICS Annex 1, you cannot use this method, even though it's very attractive in many cases. But if you have called something a grade A in particular, but if you're using that categorization of your areas, grade A, B, C, D, you need to be very careful about employing this. It cannot be used for grade A because in the Annex 1, it very clearly specifies that you have to take a cubic meter of air. That's a demand in that in order to call something a grade A. If you don't take the full cubic meter or larger, then you cannot call it a grade A. So just be aware, if you've used this term for your area, grade A, B, C, D, you need to be mindful as to whether or not you can use sequential sampling at all. But just be aware, it's an alternate method and sometimes very useful. So for example, if we have a 5 micron, need to sample 5 microns, which normally takes quite a bit of time in order to evaluate because there's so few events, what happens if you have a lower flow count system operating at 1 cubic foot per minute, 28.3 cubic feet or excuse me, liters per minute, normally you'd have to run at least 24 minutes to sample and verify that, to take a full amount of air and have the statistical confidence. However, if your observed counts within the first few minutes are very low, for example, if you count only one event at 5 microns in the first roughly 6 minutes, you could stop. You can declare that it will pass based on sequential method. So again, this is based on the assumption that the air is consistent at that point during the time of sampling, and that as you sample more and more and more in time, you'll still be getting the same result, and you can predict ahead of time how it will be, that you'll pass or you'll fail. Well, there is a companion section. We're talking about aerosol particle concentrations to the classification document 14644-2. Talks about monitoring. Now, it's not specific on how to monitor, and again, this is not industry specific. It doesn't relate to pharmaceutical, it doesn't relate to aerospace, doesn't relate to surgical suites. It's a space, talking about spaces where particles in the air and the control of those are of interest in order to have good results for some application. So when the 14644-1 was revised, also simultaneously 14644-2 underwent a revision, again effective as of December 2015. So this is a focus on the other side of particle counting, and this is focusing then on the monitoring side of it. What do we do day by day or week by week to look at an area and understand it's still appropriate for our use? So the title of this is Monitoring to Provide Evidence of Cleanroom Performance Related to Air Cleanliness by Particle Concentration. A long title, but fundamentally the important word here is monitoring. And we talk about sampling throughout a period of time between those formal classification efforts, which again might occur every 6 months or once a year. What's happening in between that time? That's important to know, and that's what the monitoring function is. And also in this document describes some of the concerns with the pressure and airflow in a clean space, because often those have an impact in the prevention of particles entering by pressure, or airflow in terms of the changes of air, the refreshing, the dilution, you might say, of the particle concentration due to the inflow of filtered air. So basically, this revision emphasized the need to consider monitoring strategy. Now, for most people working in the life sciences, they often have already a process and procedure set up for it. So this might seem a little bit redundant. Gee, we have one of those. We have a written procedure, and that's good, and that's true. But that's not true in all industries and all companies. So this is a reminder that, yes, it's great to have this effort for classification, but we need to be thinking, in addition to what we do for the classification in ISO 14644-1, with the idea of monitoring. And in particular, we look at the continuing flow of data over time. We want to know with a certain amount of confidence that this area continues to be useful to us. So that's the idea behind the whole -2 and what it discusses. So the flow of data over time. So basically, when we do monitoring, and the more monitoring we do, the more substantial it is, the better we can understand that the area is within our boundaries. And in many cases now we talk about risk analysis. We want to know that we can operate this area and carry out whatever process or procedure we intend, and we have sufficient enough quality of performance and consistency that the risk for product loss or failure or contamination in some way or another is minimized. And in many cases, we can look at that in a way to minimize operation cost and product loss by having effective control of the air around our process. This is a quick overview of the document in terms of the points. And we talk about creating, executing, maintaining a monitoring plan. And we start often seeing now in these discussions risk analysis. So it speaks to that. It speaks, well, if we're spending the money and effort to have a clean and controlled area, we must have some sense of it being a risk if we don't do it in that area. How critical is that? Are we trying to make a sterile drug, or are we just trying to maintain a certain level of quality of air for some other purpose? So it demands that we set forward some kind of a monitoring plan. It can be quite simple or more complex, depending again on what we see as the risk and the concerns with it. We look for that plan to be written and reviewed and approved by the appropriate quality group in an organization. We talk a little bit about looking at the data and about periodic classification in that document. So we do point back to the idea of needing classification at some frequency. So again, what we look about is risk. One of the things we talk about too is that certain regulatory agencies, so we're speaking perhaps in the pharmaceutical world of the TGA, the FDA, the EMA, other organizations that have demands for certain types of work. For example, the manufacturer of sterile medicine has a higher risk factor and is much more concerned to these agencies than, let's say, NASA putting together a space shuttle. Well, that certainly has its risk and its concerns, but it's different than what's needed in terms of a particular industry. And so these other regulatory agencies may impose supplementary policies or restrictions. And so we may need to think about how to monitor relevant to satisfying those regulatory agencies. This is the important reality of monitoring as opposed to classification. Observed values may fluctuate considerably due to factors such as, but not limited to, the number of personnel present, rate of airflow, operation of instruments, machinery, and activities in adjacent spaces. And this is really at the heart of the distinction, you might say, between classification and monitoring, in that we acknowledge that these spaces are somewhat dynamic, and it's certainly different in a space that has 3 people working versus 5 or 10 in the same identical space. We'll see different concentrations and signatures in the air particle levels. So we need to take those into account. We need to maybe restrict the number of people, control that within the capabilities of the space, and so forth. So that's a lot different mindset, a lot different kind of concern than simply classifying the area initially, be it at rest or in operation, and understanding the activities and the effects of that over periods of time, week by week, month by month. So again, a different intent behind the functions of monitoring. Also important to realize that these need to be somewhat living documents when you create a monitoring process. Think about changes either in personnel, number of personnel may be needed for procedure, for types of equipment being added, and so forth. So occasionally these documents should be reviewed. And in the pharmaceutical industry, I know it's always difficult to change documents. You have a standard operating procedures, and they should not be frequently changed, but occasionally you may need to review those. Every year or two should be an opportunity to look back and say, have we had an effective plan? Have we sampled too many points, which is often a concern. People sometimes get overly enthusiastic about their sampling demands, and so they put in too many points. And really think about that, because you're making extra effort. You're opening up questions about the whole monitoring scheme if you're sampling a large number of points in areas that don't have really much use. So really think about that. Maybe you need to add points because of new operations or new concerns. So every year or two, consider reviewing the plans. In particular, I might point out a couple of the addendums, the annexes to the -2 document. One is an outline of things to think about when you set together a monitoring plan. And the second is to look at the levels you might want to set forward for alert and action. So those are things that are a little bit different for particles than other things. There's a lot of fluctuation in different concerns. And the same is true for pressure differential in many ways, because those are both measurements that can change dramatically but yet not be of concern. For example, the pressure differential in a room can change when somebody opens the door and closes it again. And so for that several seconds period of time, the pressure differential looks, usually if we're thinking positive pressure, goes low, and you lose the pressure you've seen just a few seconds before, but yet then it returns. You don't want an alarm set for that. So even though it ultimately violates a level, if it returns quickly, it's not of concern. Similar can be true of particle counts. When you see or observe someone doing an intervention or some other activity that's expected or planned, then you might see an increase in counts for a moment or two, and then a reduction again. And that would be normal. Again, that kind of a process signature needs to be understood and needs to be calculated for or adjusted for in your alarm strategy. So this discusses that need for thinking about how you set warning levels and a couple of examples of things to look at or consider. So if we look at applying this new revision, how would it work? Let's say we had a manufacturing facility with 7 grade A rooms, each around 36 square meters, and we wanted to go ahead and do a classification. What would we need to do? Well, previously when we looked at 36 square meters, taking the square root of that, the value was 6 as a minimum number of locations. Whereas today, looking at the table, the value is 9. So we have an increase in the number of positions, and we have to do that for each of the 7 rooms. So now we have 9 times 7 rather than 6 times 7 for our number of points as a minimum. And so if we look at that and we say, well, it takes a certain amount of time to do each one, depending on the sample rate of the machine. Well, if we had a 100 liter per minute machine, it would take us 7 hours previously. Now it's going to take about 10.5 hours total for it. If we have a slower flow rate machine, then that's going to take us yet even more. So we have concerns with that in terms of the increase in hours to execute this. Again, there's a purpose behind it, to improve the quality of the data by having an increase in the number of points. So the purpose is giving us a better view of the room, but it does come at the demand of more time spent because you have more positions for a particular flow rate involved. How can we review that? Well, there's a couple ways to approach that and actually maybe consider being able to minimize that impact. First off, of course, is orchestrating the flow in an effective way and being able maybe to incorporate that into the instrument so it's a planned movement to make that more efficient in the process of moving through the various 9 points that are there under the new system, and maybe figuring out a way to minimize the reporting side of it. You're still going to have to go out and sample at each and every point. The demand then, of course, at the end is I have to create a report and file information. Can we speed that up? And the answer could be yes. So one of the things is time consuming, of course, always has been the idea of putting the information together on paper tape and creating some sort of permanent file, whether it's a PDF and or typing it maybe into Excel and putting it into Excel spreadsheet or up into a LIMS. That takes time and increases the risk of a transcription error. So one of the things that's possible is to use the electronics of a more sophisticated counter to help out in the process. Many counters today have, in essence, a small computer built into them, almost you could say like a laptop built onto a particle counter. So you're certainly taking the reading, the instrument's recording as it might have done previously, but now there's a lot more information available and a lot more information processing available on the instrument itself. So it helps in the whole movement of the of information. So eliminate the paper printout, electronically create PDF in the machine, and maybe an Excel output. That would certainly speed up the overall process. And that's possible today with some of the instruments that are on the market, like the Met1 3400, for example, provides that kind of capability and the ability to set up files and structures that could flow directly into a LIMS system. So if we look at this example and we say, well, a lot of the time we spend in working with this is taping, scanning, data entry. Could we eliminate those? Yes, with certain electronics, you can bring those tasks down to zero. You still are going to end up spending time for sampling the points. Can't minimize that aspect of it. Still have to go out and get the data set. But once you have the data set, you don't have to spend as much time crunching it. You create a report, and now you're down to a relatively short time using newer format, newer electronics on the instrument to help with the process. So this type of operation may make it reasonable to consider this type of equipment both for classification and for monitoring purposes. Well, what about the GMTs? What if I'm doing Annex 1? How does this impact me? Well, it does have some impact. It's not so serious, but it's important to realize that these two documents, the ISO 14644-1 and the PICS or EU GMP document Annex 1 are not affecting, you could say, one or another. Now, there is some interdependency, as we'll see, but it really are two separate documents. So what was done with ISO only indirectly has an impact on these two documents, but it's something certainly to be aware of. And that's because Annex 1 references the ISO 14644-1 document to determine the minimum number of sample points. So there's the effect, really. In other words, it's not that the ISO committee rewrote GMP Annex 1. It's simply that that document, the Annex 1 document, points back to ISO for certain operations, in particular minimum number of sample points. So this is where an industry-specific document, Annex 1, is impacted somewhat by that change to a generic, you could say, industry-neutral document. So again, minimum number of sample locations, as we've spoken about, we've moved from square root to the lookup chart. So when we examine that, we say, oh, gee, I'm grade A, I'm going to have to do an additional number of points because this is pointing back to ISO 1. So you're not changing that document, you're not changing a lot of things about Annex 1, but because it does reference that and says use that method to select, it will impact your process for classifying a grade A, B, C, D area. But again, you know, we touched earlier on the idea of sequential sampling, and this is an area where Annex 1 differs from the ISO, and Annex 1 says you have to take a actual physical cubic meter to classify a grade A area, regardless of anything else you might consider. And so if you're going to call it grade A, you have to take that physical cubic meter of air at each and every sample point. Also, it references ISO and 14644-1, and that calls for then the need to make sure your instruments fit the ISO 21501-4 calibration. A lot of older instruments would not necessarily be able to be calibrated to that, and if an instrument is not in good repair, it might also fail this calibration method. So this is something that, again, keeps an eye on the health and performance of an instrument in a much more effective and efficient way than any generic calibration method that might be used. So in summary, we see that these ISO revisions affect the number of sample points, and this again came in as of December of 2015, so this is in effect today, and it may change the operation in terms of time, and you have to deal with more data to record and more data to analyze. It's not an enormous increase, but it still is something to be aware of. But you will get better data. You'll have higher confidence the room will perform as you needed. And there are instruments out there that can help mitigate that impact, that can help shorten the analysis of the data. Or if you go from a 1 cubic foot per minute machine to a 50 or 100 liter per minute machine, it'll actually help save time in the classification process. So again, keep in mind that even though we talk about particle counting in general and just think about air concentrations, when we speak to classification, we're looking at these documents, the Annex 1 document and the 14644 document, and when we're talking about monitoring, we're really looking at your decision how to monitor it. It's your SOP, you could say, your monitoring plan that defines that. It's not defined by ISO 14644-1 or Annex 1 when you do your daily, weekly, maybe even continuous monitoring. You're looking at your needs, the risk that you see in operating your particular facility or area. So with that, I'll wrap up the discussion point, and we'll turn to the questions that are there. Again, I want to thank you for your attention. Here's my email address if you wish to have a contact. And also, I'd like to call your attention to some forthcoming events. In the United States and North America, we have our Life Science Users Group events coming up. These are educational seminars that are held at no cost in various cities. In the U.S., in May, those will be held in Berkeley, California, Irvine, California, Bloomington, Minnesota, in Malvern, Pennsylvania, and Research Triangle Park, North Carolina. That schedule will be found on particle.com, posted in the near future. I don't believe it's up there quite yet, but you should be able to go to particle.com and find that information out if those are areas where you can you're located or travel to. Also, there's some other educational webinars that will be coming up on LabRoots on the same platform. So keep an eye open for these. We'll talk a bit more about ISO 21501. There was a discussion too coming up about compounding pharmacies, and also previously recorded one for the USP 787 and looking at particulate matter in liquids in therapeutic protein injections. A discussion of that, and there'll be ongoing subject matter available. So keep attuned to the offerings at LabRoots relative to particles, and that may be helpful in your daily work. So let's go over and look at questions that have come up on the discussion panel here.
That was an excellent presentation, Joe. Thank you so much for bringing that information to us. Before we get started with all of your questions, here's a quick reminder about how to reach us today. Questions can be sent via the green Q&A button at the lower left of the presentation window. We get to as many questions as we can. We have some great ones coming in right now. But while questions are coming in, we would like to conduct a poll asking you, how likely are you to recommend this webinar to a colleague? We would really appreciate your answer to this question. So while the audience members are answering the poll, I'll ask the first question, which comes from Mark, who asks, under the new revision, How many counts, how many locations, and for how much time would you sample a safety cabinet work zone that had an internal volume of 0.6 meters cubed?
Okay, the chart is set up for something obviously larger than that, and there's many certainly small cabinets and areas, biological safety cabinets, just very small operating areas that fit under that. So you would go to the first entry in that chart, which was a maximum of 2 meters squared. And that's the one where you would use it. So you could technically fulfill that with a single point and a single sample. And under the new regulation, you could classify with that. And that would be all that is required under that under the new regulations.
Oh, that's great. Our next question comes from Dominique, who asked, our question concerns the new ISO 21501-4. What about concentration limit measuring during calibration?
Generally, concentration limit, and I think Dominic's probably looking at the maximum level, is something that doesn't need to be tested at each and every 501-4 calibration in the field. Certainly it's something that's evaluated in the design and the manufacture of an instrument and tested, but generally it's something that is will not be changing in an instrument over time, because it's fundamentally based on the physical space of the detection area in the sensor, and that really will not change radically. If it might happen to, there's other aspects of the calibration method will pick up some radical disturbance of that area. So generally in the field, that particular function is not tested.
Okay. Okay, our next question comes from Andreas. It's a little bit long, so feel free to read along with me, Joe.
All right.
Is there a special text passage in the EU GMT Annex 1 that tells to measure 1 meter cubed in Class A? And then he writes, forbidden to take the sequential method. I think it is only given by the ISO 14644 Class 4.8 formula limit of 20 particles of 5 micron and the formula 20 per class limit times 1,000, which is the minimum volume per measuring point.
Right, and it is, your point is well taken, and exactly as I was trying to define earlier. Sequential sampling is very useful in a lot of instances. However, it is not something you can use for grade A under the Annex 1, and that is because it is specified in section, I believe it's section 5. I'm not 100% sure, but I don't have it in front of me. I think it's section 5, it could be 6, but somewhere between 4 and 7 is the discussion in there that a full cubic meter needs to be taken for a grade A area. It also works out when you look at the formula nowadays, since that limit is at 20, the formula basically also would indicate the need out of ISO to take a cubic meter if you had a target of 20, but that's incidental. So it's really, if you're going to call an area grade A, you have to abide by that classification demand, and that would indicate you must take a cubic meter, or at least minimum cubic meter for anything if you're going to therefore later call it a grade A area.
Okay, great. Next we have a question from Leslie, who says, having just completed classification validation of a number of clean rooms using the old standard in June 2015, will this validation need to be repeated using the new standard 14644-1?
Sure, good question, and I think one that's on a lot of people's minds. There's no demand that you would repeat it immediately using the new standard. Again, we tried to make modest changes in it so that the classification of rooms are unlikely to change. Assuming the room is kept in good order, that's probably quite true. What it means, though, is that the next time a classification is done, 6 months or a year from now, you would go ahead and use the new standard and use additional points as required. to execute a classification, and you would say, yes, I classified according to 14644-1 colon 2015, as opposed to what it is now, which is colon 1999. That's a date code there, right? So it's a year reference. So you just say, I now classified according to this, and it would simply mean a few additional points, and ideally and probably true, it will not change the classification of that area or room. But it just means you have more confidence because more points are taken. It's always true. The more points you take for a statistical effort, the closer you approach full confidence. So again, the intention here was to bring the old standard up to the point where we could say, based on the data with 95% confidence, this data set indicates the whole room is ISO class, whatever it might be, X. Okay.
Great, thanks. Next we have a comment from Sandrine who says, do we have to do monitoring? Is it for certain ISO classes or for some activities?
Do you have to do monitoring? No, you have to have a monitoring plan if you're going to have kind of an ISO area. In other words, you say, oh, I have an ISO class 8 room, and I'm doing some very low risk work in it, but I am calling it ISO class 8, and once a year I bring a particle counter in or have somebody come in and test it. Do I have to keep monitoring throughout the year? No, there's nothing that demands that you do that. It's a matter of wisdom based on your sense of risk of the area and how changeable you feel that area is. So ISO doesn't demand that you monitor. It does demand that you have a plan in place, which could be saying, I'm not going to monitor, and here's the reason why, and it could be simple as that. But it simply says you do need to put that intention in writing, and it could be extremely simple. It could be saying, I'm not going to do it, or I'm going to only do it once a month. That's fine. But there needs to be something put forward, and that's what the ISO 14644-2 is pointing to. You need to have some kind of stated plan and follow it. And that, again, could be simple, and it could even be a statement, we're not doing anything in between. So there's not a, so to speak, gun to your head to monitor. There's nothing that states a frequency. It's really based on how you want to use that place and how risky you feel it would be to not have any understanding of that area throughout a period in between classifications.
Great information. Next we have a comment from Dilip who says, great presentation. And can you explain that the conflict between ISO guidance on frequency of certification? Specifically, he wants to know about the FDA guidance in Annex 1 that requires every six months, which industry considers to be the worst case. Please let us know what you think, what your personal opinion is.
Okay. Another good question, because there's a lot of concern, again, between what's asked for by regulatory agencies and what ISO is pointing to. And again, ISO is industry neutral. It doesn't think about what's happening in the space. It just says if you want a statistical method for looking at or assessing the particles in a zone in the air, here's a method. When you come to certain agencies, be it NASA, be it a surgical suite, be it sterile drugs, there are agencies such as the FDA and EMA that mandate certain types of operations, and they mandate something differently for aseptic operation of a sterile injectable versus ointments or creams. So they have different approaches depending on what they see as risk. ISO is not going to come forward and put a specific time period on as they had before, because it's really significant and relevant for a regulatory agency to put that forward. And if the FDA and EMA want to say for a aseptic area, it should be classification every 6 months, that's not without some logic. There should be, of course, a general monitoring program in those higher risk areas that watch for day-to-day variations. But it's not a bad thing every once in a while to step back and take a more formal look, a more thorough look at a complete space or environment, even areas that are not typically in a day-to-day manner used, where you're not really having a risk factor for it. It's still good to look at the whole area on an occasional basis. What occasional might mean is, again, back to the regulatory agency to set. And if they say 6 months, okay, that may make sense. Some other groups might say, hey, every 24 months is fine. So again, it's really a matter of understanding any regulatory demands that are specific for your application. And 6 months in a case where you're dealing with aseptic manufacturing, to me, makes pretty good sense. You know, but with the monitoring program, you would be looking really for changes in very key and high critical zones. And that again back to the difference with monitoring and classification. Because classification is just a momentary snapshot. It doesn't look at the dynamic characteristics of the room, impacts of changes in the airflow due to changes or failure of a ventilation system, filter failures due to various different causes, even including, let's say, earthquakes upsetting the seals on the edges of filters. All those things happen, and classification may catch those, but again, it's done in a very infrequent basis. But it is a momentary, more thorough study, and therefore of use, and it's up to that particular regulatory or control agency, as ISO is set today, to put that time period.
Great. Next we have a question from Ayo, who asks, in pharmaceutical clean rooms, would it be required to reclassify our rooms, especially where the number of sample locations required is now higher?
Generally, the answer would be no, and you again only approach this the next time a classification effort was needed or specified for that particular room. You shouldn't have to reclassify it until you come up to the next date, which might be 1 year or so. And again, ideally, even adding the number of points shouldn't change your classification unless there's some unusual aspect to the room or zone. So I wouldn't expect the classification to change, and it just again gives you a few more points and statistically gives you a little firmer ground to stand on when you make your statement, yes, this is an ISO 5 room at 0.5 microns. So that's the intent again of adding the points, is just to improve the statistical foundation of that statement.
Great. Next we have a question from UAP, who asks, the number of sampling points, whether the number of sampling points should remain the same for classification and monitoring, or can it vary?
Oh, definitely can vary. And this is a mistake I think a lot of people tend to make, and maybe even have inherited as an operating procedure from someone else. They say, oh, we had these many points for classification, and therefore we need the same number for monitoring. And generally, that's not at all necessary. And in many cases, monitoring involves significant reduction of points versus classification, because generally monitoring is looking at a particular operation or operations in a room, and it might be a large room, and the operations might take place in only 1 or 2 square meters out of the whole room. And that's what you want to focus on for monitoring. Where is my exposure? Where is my risk? Where do I want to have some understanding of what's happening in the room? So in many cases, the number of points used for classifying will be markedly different from those used for monitoring. But that's really a process-by-process sense, and it's something that isn't specified anywhere. But certainly look at the amount of time and the information gained by your monitoring methods. And in many cases, people find that they're over-monitoring, you might say. They're taking a lot more points, and they're not using the information. And that's kind of the key to it. If you're recording information just because historically everyone else has done it that way, but if you look at the data, and especially if you have data that fluctuates and nobody's acting on it, in other words, there's no actionable method. It's like, oh, don't worry about that point. It always goes high, at this time, and it doesn't affect anything. Stop taking data. Look at that again as a part of an annual or maybe biannual review every 2 years. Look at your methods and say, does this still make sense to be taking this point, or even should we add them? But in most cases, the number of monitoring points will be a small subset of what you do for classification.
Okay. And along the same lines, Nilep has a follow-up question asking, are these requirements only for clean room or non-sterile manufacturing areas?
For the ISO, so focus or be specific about the ISO. The ISO 14644 speaks to any room where you want to control the particle levels in the air. So sometimes people use clean room in a few different ways. It's a matter of what's been needed in terms of clean room. But if you're talking about the air in the clean room and the particles in the air, the concentration of it, this can be used for that. And it's not a matter of it being sterile or not sterile. It's a matter really of, do I want to control the level of airborne particles? And this is a way to analyze or understand that you've got a base that you can use for a particular application. And the application, again, is not something ISO 14644 takes into account. Now, Annex 1 very much takes that into account. It says, you're doing something where you are supposed to have a sterile product, you do this. Okay, and that's fine. That's what one industry asks for. ISO, again, is industry neutral, and it's just a method to use for evaluating a space where you're wanting to understand the concentration of airborne particles and determine, does that meet my needs? So again, it's important to realize ISO is industry neutral. It doesn't think about the application. You could be painting a car. You could be doing surgery. It doesn't think about that. It just simply says, here's a way to do it, and something that will give you 95% confidence if it's done correctly. And that's the end of that subject. And anything beyond that, whether you're doing sterile, non-sterile, ointments, anything else, is a province of a regulatory demand or GMP, and not from ISO.
Okay. Next, we have a question from Jeff, who asks, the prior version of ISO 14644-2 required reclassification of ISO Class 5 to be done every 6 months. That table in Table 2 has been removed. Is that still the expectation for pharmaceuticals ISO 5 every 6 months? Or would 12 months now be followed based on the Section 5 of current version of ISO 14644-2?
Well, without knowing anything further about what's being done in the room, you would say, I can go to 12 months. The question that has to be asked is, what's being done, and has a regulatory agency set forward a particular constraint? Because you may find that a regulatory agency says, every 6 months must be followed. That's a decision that is based on the GMP, not on ISO. ISO is probably, you could say, the larger set or the more adaptable classification document. But then you get to specific needs, such as Annex 1 or the FDA's sterile guidance, and you find a particular direction for a particular application. So without knowing the particular use, immediately you could say, I fall back to what it is currently, that is 12 months, and I can relax my classification. However, you may also be under the remit of a regulatory agency that says, no, that's not quite sufficient for the risk involved. We want it done more frequently. So ISO, again, is not looking at a particular application, whereas certainly the regulatory agencies in their various guidances and GMP put forward very specific demands on users of clean rooms.
Okay, and as a follow-up, Jeff also asks, is the optional test for airflow visualization, filter leakage, recovery, etc., listed in the older version of ISO 14644-2, Annex A, going to end up anywhere? He says he doesn't see it in the new versions of either ISO 14644-1 or 2.
Okay, good question, and if you've been in the industry a while, it's probably something that might pop into your mind. It is going to end up in the -3, forthcoming -3 version, so it's being ported over to a different 14644 document. So for the meantime, you could use what had currently or had existed and might be in your monitoring plan. There will be some future release of -3, I believe, probably within the next year. I'm not in that committee, and I don't know precisely, but I know it's definitely ongoing work. And you should see some definition there. I don't think they're going to radically change. So, I don't expect any shift of that recommended further testing for airflow and visualization, but that'll be in -3. For visualization, that's something that's very important for regulatory agencies. So if you fall under their province, then dynamic visualization, especially of sterile fill areas, is much in the interest of FDA. They've written some 483s related to people not understanding how the room works under dynamic conditions and in high-risk operations. So keep that in mind as a separate topic, but that's not ISO. That's not 14644 again. That's specific to GMP and to a particular regulatory agency. So high-risk operations, visualization is very important, and not just static, but dynamic too.
Great information there. Next, we have a question from Federico to ask, the particle limits that you showed on the table, are these non-viable particles, viable particles, or both?
I suspect the proper answer you could say is both. It isn't specific, and it's basically generally considered in that table to reference from the pharmaceutical world non-viable particles. But truthfully, I think a better title is total, in that anything that's in the air, be it living or not living, or be it viable, that is able to reproduce or not, to the particle counter and to the table is not significant. In other words, a particle counter sees a organism or a skin flake with a bunch of organisms on it as a particular solid entity in the air. It doesn't think of it as viable or not, and it just says there's one of those, it counts it. So the table is really, you could say, non-viable particles, but it's also, I think, a better phrase to have called it total. And if you notice in the document, it doesn't use either phrase in discussing that in the ISO document. It just says airborne particulates.
Okay. Next, we have a question from Dominique who says, what tests have to be performed during calibration?
Okay. And I'm going to guess this is probably looking at 21501-4 in particular. And again, there's a difference between what's done in production or initial design versus what's done in the field, because some tests, doing them in the field, are limited. But if it's a 21501-4 type test, you're looking for, size definition. You're looking for verification of the volume sampled either by a airflow test plus time, because you're always reporting counts per volume, so you need to verify that. And then you also want to look at your counting efficiency at a couple of different sizes as specified in the document. Those would all be things that are critical to the to fulfilling a 21501-4 type calibration. And that's something that I could handle separately with Dominique here, and we will be having a further, more detailed discussion of 21501 on a LabRoots seminar in the next couple of months. I don't think there's an exact date, but that's will be a forthcoming and more detailed topic.
Okay, great. Well, we have time for one more question, and that question is from Jeff, who says, if I classify an ISO 5 grade A area with 10 locations and routinely monitor 4 locations based on risk, is the expectation that I reclassify every 6 months by taking those 10 locations, or is the frequent monitoring being done routinely at 4 locations enough to offset that, and the reclassification is not necessary?
Okay, and this is where, again, that choice of the designation of grade A creates a different demand than just a simple ISO 5 statement. ISO 5 alone would allow you to consider continuing operation based on good data out of those 4 positions, but grade A would not. Okay, grade A demands that you go back and you classify on a more frequent period and all points. So you can't shorten that period, or you can't use that data if you're calling it grade A. And so this is an important distinction. And again, a focus on something that as ISO 5 would be industry neutral and not thinking about what you're doing. Whereas once you say grade A, you're pointing to a particular function that is manufacture of sterile medicines that is unique and has its own demands. So unfortunately, if you are calling it grade A, you're going to fall into the demand that you do all 10 points on that more frequent basis, and you can't avoid that, or you have to change the designation. You can't call it grade A.
Okay, well, that was fantastic. Thank you again, Joe, for bringing us this presentation. Do you have any final comments for us before we go?
First off, thank you, and thank to everyone there at LabRoots too for conducting all this, putting it together. Thank everybody for attending. It is a interesting and kind of confusing topic, and hopefully today we've made a little progress, especially in this distinction between classification and monitoring, and to define how each one can be used, even again, as we say, often using the same instrument. But these topics are important, and keeping control of an area at an appropriate level is something that monitoring can help us with. And then the less frequent but more formal study on a classification helps everyone have the sense of confidence that an area will perform in the way that they need it to execute their work. So again, thanks everyone. Certainly we'll be happy to take other questions in. We'll try to answer these by email. So again, thank you for your attention, and appreciate your time today.
Yeah, that was terrific. Thank you, Joe, again for bringing this presentation to us. And special thanks to Beckman Coulter for sponsoring this event. And before we end, I just wanted to let you know that today's webcast will be available for on-demand viewing through August of 2016. You will receive an email from us alerting you when it's available for on-demand and posted on labroots.com. Feel free to forward this announcement to any colleagues who weren't able to join in today. Thank you again for logging in and participating in today's broadcast event. Hope to see you next time. Bye-bye.